Good afternoon, everyone. Welcome to SensorSRIG's Spring 2021 conference series. This is our second seminar presentation this semester. I'm Aminur Rashid Chaudhary, moderator for SensorRIG conference series. And the SensorRIG is a multidisciplinary group of researchers with interest in sensor materials, methods, devices, and sensors, applications for healthcare environment, civil, petroleum, chemical industries, and military applications. This group provides an opportunity to exchange ideas, present early findings, and explore collaborations with interdisciplinary funding opportunities. Sensor RIG's aims are to connect sensor technologies, middleware, and applications, and to provide an interface between academic and industrial research. Today, our presenter is William. Let me introduce William to our audiences. William Meador is a fifth-year graduate student in the Department of Biomedical Engineering at UT Austin and a NIH Ruth L. Christensen postdoctoral fellow. In 2017, he joined Dr. Manuel Rauch's research group. His research interests include soft tissue, biomechanics, medical device design, and cardiovascular system. William plans to submit his dissertation in fall 2021. He previously completed his Bachelor in Science at Case Western Reserve University in 2015 and his MSc at UT Austin in 2019 in biomedical engineering. Outside of academics, William is an avid golfer, a former college basketball athlete, and aspires to visit every US national park. We wish good luck, William, on that. And before giving floor to William, I would like to express my invitation to all interested personnel to join SensorSRIG Student Committee as executive members. We would be very happy to have you in the team. SensorSRIG give you an opportunity to utilize skills and make network with other interested personnel. So, thank you. I would like to give William floor to present his research.
Great. Thank you very much for that kind introduction. I will share my screen. Let me get this going on my end. Okay, can everybody see my screen and hear me? Great.
Yes, we can.
Perfect, thank you. So yes, thank you for the kind introduction. My name is Will Metter. I am a fifth-year PhD student in the Biomedical Engineering Department at UT Austin. I work with Dr. Manuel Rauch in the Soft Tissue Biomechanics Lab. And today's talk, I'll be talking about the unique challenges we face in characterizing cardiovascular soft tissues, specifically with regard to the biomechanics field. In the interest of the many different talents that we have in the audience, I will be keeping this talk fairly general in hopes that it fosters future collaborations and that you find your own particular interests within this discussion as well. So to give a brief overview, just starting from the beginning, what are soft tissues? Soft tissues are tissues that connect and support and protect the structures and organs of our body. Most often thought of as muscle, tendons, cartilage, ligaments, anything not bone in our body is a soft tissue. And so as biomechanists, we are interested in characterizing these soft tissues because these tissues are alive, and as we age, as we get injured, or as we get sick, they adapt, and we want to be able to capture this. And so it's interesting not only from a basic science standpoint, but also a clinical standpoint with the introduction of medical therapies for injured soft tissues or diseased soft tissues, as well as predictive and diagnostic modeling or simulations. Our work is, as a lab, is very heavily, as the topic of my discussion suggests, it's very heavily involved with the cardiovascular system. Specifically, we are interested in the lab as a lab on what's called the forgotten side of the heart, which is the right side of the heart. It's referred to as the forgotten side because there's relatively less research on the components of the right side of the heart. So looking at the right side of the heart, we have the right atrium. and the right ventricle, which are the two chambers of the right side of the heart. And these are spanned across by the tricuspid valve. The tricuspid valve is a 3-leaflet valve circumscribed by a D-shaped fibrous annulus. And in the subvalvular apparatus, we have chordae tendineae spanning between the leaflets and the papillary muscles along the endocardium of the right ventricle. Now, the tricuspid valve is what I will be talking about very heavily today, because that is the topic of my dissertation. And the leaflets themselves, the valve, it is responsible for regulating blood flow between the right atrium and the right ventricle. And the important aspect of this is that the blood flow is unidirectional. So it acts as a check valve, and we only want blood flow going from the atrium to the ventricle. And as we can see, there's 3 leaflets, anterior, posterior, and septal, that come together to coapt to prevent that backflow of blood. Now, the disease with this backflow of blood that's associated with it is called functional tricuspid regurgitation. Regurgitation just simply means the backflow of blood, and it affects 1.6 million Americans in a clinically significant manner. So in 85% of these cases where we have tricuspid valve failure, it's tagged with this clinical term functional. And what this functional term means is that it implies that the valve itself is healthy and intact, but instead valve extrinsic factors are causing it to fail. And so what this looks like within the heart is we might get a dilated right ventricle, which displaces these papillary muscles, tethers the chordal attachments to the leaflet, and therefore they're unable to move or coapt. Similarly, a dilated right ventricle can result in annular dilation, which simply just puts the leaflets further apart from one another and therefore can't coapt, and then we get backflow of blood. Now, there are treatment options to patients that have this disease, but we view this potentially as being suboptimal, because in just a few years of surgery, up to 30% have recurrent regurgitation. And so as an engineer, we look at this and we think, okay, there's something missing in the way that we're approaching this treatment of functional TR. And so the current paradigm, if we look at this, and this is what our lab is very interested in, is that this functional tag suggests or implies that the leaflets themselves have this passive nature, that they aren't involved in their own disease. But in reality, we know that this tissue is alive, it's mechanobiologically sensitive. And so what we instead are wanting to investigate is this paradigm that the tissue itself could be active and playing a role in its own disease. So in order to do this, we work with some clinical collaborators out of Spectrum Health in Grand Rapids, Michigan, and they developed this really helpful ovine model in which we can compare healthy sheep, so control sheep, to this tachycardia-induced cardiomyopathy model. And this is a big, long, scary word that basically says we rapidly pace the hearts of the sheep to induce heart disease. So TIC for short. And what's important is we're able to reproduce biventricular heart failure within 3 weeks with these animals. And it's the clinical manifestation, meaning that in medicine, we rarely just see clinical tricuspid regurgitation. It's usually with comorbidities like reduced ejection fraction, biventricular remodeling, and the other atrioventricular valve, the mitral valve, is also regurgitant. So through using the tissue, we can investigate whether the tissue itself has an adaptation to the introduction of disease. And so we take these tissues and we approach this from a biomechanist standpoint. And so the field of biomechanics really bridges the idea that a tissue's function is directly linked or bridged by understanding what the structure of that tissue is. And so we have these tissues, and then we experimentally test and expose these tissues to different assays or experimental methods to try to identify the function and the structures of these tissues. Now, this in our field is by no means a simple task. These tissues have their own challenges, which I'll go over, but this is a very simplified version of what we do every single day. But this is the overall attempt that we're trying to identify function and structure and bridge those together. So to go over a couple of the challenges of cardiovascular soft tissues, if we do mechanical testing, we'll see that they have very complex mechanical properties. So this is a stress strain curve, and this is a very classic J-shaped curve that we would see in the field of soft tissue biomechanics. for collagenous materials. So we get nonlinear behavior. If we test it in two different directions, we see different behavior depending on where we stretch it or which direction we stretch it in. We call this anisotropy. We also do time-dependent experiments in which we can see these time-dependent effects of stress relaxation, viscoelastic effects, etc. So very complex mechanical properties that we must be able to identify. Second off, these tissues are biologically active and adaptive. And the most simple case of this in the cardiovascular system is a hypertrophic heart. A hypertrophic heart can happen with even physiological conditions or pathological conditions. So athletes of the elite status are known to have athlete heart, which is a hypertrophic growth of the heart, but also you can have hypertrophic cardiomyopathy just from having cardiovascular disease. So again, these tissues are active and adaptive, and depending on who you probe the tissue from, it'll look different. Again, to reiterate, these tissues are made up of many different structures, and they're at many different length scales. And so it's not just to say that this is tissue. It's really the buildup of many different things, down from the molecular and cellular scale all the way up to the microstructural and the tissue and organ scale. So these are all very complicated structures that we have to go through. And then just a courtesy blood warning for anyone who is squeamish, now would be a good time to look away. But these are electrical, chemical, and mechanical organs. And so when we remove these from the body, we kind of take to investigate them, we kind of take out this coupling that was actually native to the in vivo state. And so this is actually a very big challenge that to understand how it actually is within the body. But we can really only look at it outside of the body. So this is another challenge. And lastly, the concept of heterogeneity. So this is a tricuspid valve leaflet, and you can see it has a very clear layered structure. But depending on where you look within the leaflet, it's going to have very different constituents in different shapes, sizes, and everything. And this is just one type of tissue within the heart, and there's multiple different types. So again, heterogeneity makes it very complicated. So with all of these different challenges, as engineers, it requires us to develop equally sophisticated methods of sensing so that we can track these properties and then make statements about what we know about these tissues. So again, as I mentioned previous, I'm splitting up my discussion in terms of tissue function and tissue structure. First, we'll talk about the tissue function. So we use a method that we refer to as in vivo biomechanics. In vivo biomechanics simply refers to within the body. And so our collaborators at Spectrum Health implant these sonomicrometry crystals as marker positions within a tissue itself. Once these are implanted, these are piezoelectric crystals that are able to sense where the relatively the position of other crystals are. And so throughout the cardiac cycle of the heart beating within a sheep, we can sense where these markers are and therefore understand where the tissue is. What this allows us to do is to get in vivo leaflet strains throughout the cardiac cycle. And so just to compare, between the control animals that I discussed and the diseased animals previously that I discussed, we see that in vivo, we have reduced strain with disease. And so this is very, very helpful to understand and elicits that these tissues are experiencing different mechanical environments in disease, so therefore would likely be responsive. The second step to this is to do what we call in vitro biomechanics. And so this is when the tissue is shipped to us here at UT Austin and we do experimental techniques on it. The most common experimental technique that we do for these leaflet tissues is because they are of a membranous type, we can do biaxial testing. And so biaxial testing refers to doing testing in two directions simultaneously, each with independent load cells and motor control. And then we can perform through the use of optics, digital image correlation to track local strains within the tissue. What this allows us to do is to quantify some metrics of it's very difficult to quantify the metric of a nonlinear curve. And so what we can do is we can take the stiffness at different areas of these curves and quantify and compare between groups. So here we see that between control and diseased leaflets, we do get a different characteristic path for the stress strain curve. And what this results in is when we analyze it, we see stiffer behavior in the radial direction, specifically in this upper region of the curve. And then we also get stiffer behavior in the circumferential direction in the lower region of the curve. So again, the in vitro biomechanics agrees very well with the in vivo biomechanics in that these tissues are stiffer and therefore will not strain as much as easily. So very interesting findings in terms of the tissue function. But we also want to investigate the tissue structure. And this approach often revolves around using imaging techniques. And so the first imaging technique, so you'll notice a lot of the sensors are optics, but the first imaging technique that I want to discuss is the use of two-photon microscopy. And if you're not familiar with two-photon microscopy, it's a very ingenious technique, very complicated. But the two-photon microscope gives us the advantage of seeing deep within a tissue. And so we can take Z-stacks of images throughout tissue that's up to close to 1 millimeter thick, which is very fantastic and elite. So through the use of optics and these photomultiplier tubes, which can detect even singular photons, we can create really beautiful images like this. And so this image is of in green, we image collagen using a non-optical property called second harmonic generation. In blue, we have these nuclei, which we stain for. And then in the background, you can see some faint red. This is elastin, which is an elastic protein, and we image this using autofluorescence. So this is very adaptable and very useful method that we use. And when we get Z-stacks of these images, we can take pictures of each constituent and then characterize them structurally. And then we can say that at any given depth throughout the tissue, we can characterize what orientation collagen is in terms of a probability distribution function. So very useful. And in our study between control and disease, we were able to detect changes in the collagen microstructure. And specifically, we saw dispersion in the atrialis, which is the uppermost level, the side of the leaflet that faces the right atrium. And we saw dispersion of this collagen. So the collagen is adapting and changing. And so these tissues are active. The next technique we use is just through light microscopy. We stain these tissues with different histological stains to assess their constituents, as well as performs immunohistochemistry on very specific cellular markers. So with the histology in this study to determine, we were able to get a spatially resolved thickness map of these tissues as it goes from an annulus to free edge of each leaflet. And we see not only is it heterogeneous along the length, but with disease, we see this increase. And so with disease, tricuspid valve leaflets are thickening. And so this was a very interesting finding as well. And so next, we wanted to look at immunohistochemistry. And immunohistochemistry is usually you target a specific cellular marker. And so for this, we were interested in markers that are indicative of a profibrotic or remodeling state. So here we use alpha smooth muscle actin, which is indicative of valvular interstitial cell activation. And again, we can acquire these really cool spatial heat maps to see what parts of the tissue are active and which are dormant or underregulated. So this is a very useful technique, but for example, in this image, we see that along the atrialis surface, we have much more activation of valvular interstitial cells. The last of our techniques is a biochemical assay. And what we use for these is to acquire quantitative data of the constituents that are within our tissues. Some may know this as a plate reader, a spectrophotometer, but essentially what this is that you do a colorimetric assay, and then using the absorption spectrum of these assays, compared to a standard curve, you can acquire quantitative data about how much collagen is within these tissues or any other constituent that you want to assay for. So we use collagen, so it's a hydroxyproline assay. In this, by taking samples from different spatial locations within a leaflet, we can acquire these same maps that tell us where collagen is either increasing, decreasing, etc. So in this particular study, when we were doing control versus disease, we saw increases in the near annulus region, the free edge region, and almost significant in the belly. So overall, increased collagen content in these diseased leaflets. Now, to just summarize some of our findings in brief, in these leaking tricuspid valves, using the methods that we use, the sensors and biomechanics, we found 1.3 times larger leaflets with an increased thickness, which contain 1.4 times more collagen within the leaflets, and this resulted in 1.3 times stiffer behavior. All of this can be found in much more detail in our most recent publication in eLife. If this type of work interests you, be happy to discuss it outside of this in more detail as well. But to kind of allude to some of what our data is going toward, I wanted to include some simulation data as well. So this is work of our student Mrudong Mathur, who works on implementing a lot of the data that we collect in the lab into a computational model. And this is for patient-specific modeling, and he can even use the data that our clinicians use back in Michigan to inform the boundary conditions of these finite element models. So very interesting applications for this data. And again, another fun thing out of Mrudong, he's very good at creating these things. If you want to see a tricuspid valve in your home, he set up this augmented reality. I'll come back to this. You can scan the code on your phone and you can put a tricuspid valve in your living room if you're interested. So very cool stuff. But overall, I want to say thank you for everyone's attention. I'm especially thankful for my lab mates, Mrudong, Ella, and Satirius, as well as our collaborators out in Spectrum Health, and of course, our funding that makes this research possible. So I'm now open to questions, and I will go back to this so you can scan the QR code if you are interested. Thank you.
Thank you, William. This was really a nice presentation. I didn't know that so many things can happen inside my heart. So this is really nice presentation. And yes, Dr. Manuel Rest, actually, yeah, we have been mentioning the thing about you since the beginning of this presentation. So I'm definitely sure that William is really thankful to you. And now I would like to give floor to our audiences to ask any questions or anything.
Thank you.
Please feel free to unmute yourself or send the question through text.
Hi, Will. I'm EJ. I work in the Office of the Vice President for Research, and I support research interest groups. Amazing presentation. I hope you go into academia and teach. I mean, half the things I didn't understand what you were saying, but I felt like I was following you, which is an amazing skill.
Appreciate that. Thank you.
I wanted to ask about, so this is a really cool application and it just seems really fun, but the modeling application that you showed us before the slide, what can that be used for? Like, how can clinicians use that to help them with their patients?
Absolutely. So obviously these types of simulations require a lot of data that we get outside of the body. And so in an ideal sense, we could capture a lot of these data by using non-invasive imaging in a clinic, and then could be used for predictive diagnostics. So what Mrudong is specifically interested in implementing with these models is implanting medical devices to see, predicting how beneficial this could be for a specific patient's tricuspid valve. And so we can run these simulations to see, there's different types of devices and sizes that you can do. And so we can predict that one device's promise over another is, I believe, what would, I don't believe we're here yet in terms of the diagnostics, but that's kind of what the aim is of doing these simulations. Simulations and modeling also reduce the need for animal models in the future. By collecting the data and implementing these into computers, this is obviously beneficial from an ethical standpoint as well. So,
well, speaking of, was that little GIF of the heart an animal heart, I assume then?
So this is actually a human tricuspid valve, I believe.
Oh, the where the heart was pumping, that image in one of
your in the, okay, the video, the bloody
yeah.
video. Yes. So that was a donor heart that was rejected. And so that was kept alive, and that's actually how we acquired a lot of the data for these human tricuspid valves as well. The data I presented was ovine. That was a donor heart that obviously that's a very valuable specimen that we were able to collect the data from. So we do have some work in our lab on human tricuspid valves as well.
That's really cool.
Thank you very much.
Hey, EJ, I want to be very clear. I 100% agree with you. I've been trying to convince Will to become a teacher and stay in academia. We're still working on it. We're still working on it. I have 9 months left to do so. So I really appreciate you reinforcing that.
It's a very hard thing to understand and know what you guys are doing, but to teach it is, you know, hard to come by. And I'm sure a lot of people have experienced that, where you meet wonderful mentors, but they're not so good at the teaching portion in the classroom. So when I see those people, I have to say it.
I appreciate that. Thank you very much.
Yeah, and also I'm going to make a plug. If you don't see your profile in our SensorsWork gallery, please complete our form so that we could get you added in there and connect you with the rest of our UT Sensors committee. Let me find the form link and I'll put it in the chat.
I think this is like really nice presentation. I have these videos and all those things that they're really nice.
Thank you.
So if no one has any questions, I would like to thank William and all our audiences for attending these beautiful presentations. And please feel free to send any kind of questions or suggestions regarding this presentation or any presentation or related to SensorRigs to our Twitter handle or our email at sensorrigsstudents@utlist.utexas.edu. And yeah, and I would like to invite all of you to our upcoming presentations by Dr. Dmitry Gaidev. And if any of you have missed the presentation, this video, this presentation will be available on YouTube and you can check it over there. You can subscribe to our YouTube channel. Our YouTube handle is also SensorRig. Please feel free to check over there. Thank you, everyone. And hopefully, William, good luck on your research and good luck on your graduation.
Thank you for the opportunity. I appreciate it. Thanks, everyone.
Thank you, Will.
Thank you.